Welcome to the NCOH monthly webinar. My name is Maarten Hoek, Communication Manager for NCOH and your host for today. This meeting will be recorded and made available through the NCOH website on our YouTube channel. This month we'll have presentations on COVID-19 and companion animals. Before I give the word to the guest today, just a reminder that you can ask questions using the chat function. So let us start with the presentations. Our first speaker of today is Professor Arjan Stegeman. He is Professor in Farm Animal Health at Utrecht University, where he is about to celebrate his 21st year work anniversary. His research focuses on the epidemiology of infections in farm animals, and research questions that keep Professor Stegeman awake at night are SARS-CoV-2, antimicrobial resistance, horizontal gene transfer, and avian influenza. Professor Stegeman will tell us more about the project fighting COVID-19 in animals and humans, the why and the what about this project. Professor Stegeman, the floor is all yours.
Thank you, Maarten. I'll try to share my screen first, so if you can tell me whether that has succeeded, then I can continue.
Yes, that seems to work. Maybe you can make it large. Perfect.
Okay, thank you. Yeah, it's always a bit doubtful when you celebrate your 21st year at the university, because it means you're getting old. But on the other hand, it was a very rewarding time. So and still, of course, our government is increasing the age of retirement constantly. So there's still many years ahead of me, I hope. So it's my privilege to give a kickoff of this webinar with the why and what's of the project fighting COVID-19 in animals and humans, a project that was started in May last year in the Netherlands, just to investigate the role of animals in the transmission of COVID-19. And well, why would you want to do that? We all know that SARS-CoV-2 originates from an animal source. We've well, at least people expected it is the horseshoe bat, maybe through a kind of an intermediate host that is still not elucidated. We have all read in the newspapers and seen on the news that the WHO committee visited Wuhan a couple of weeks ago, or actually just returned. And I think as an NCOH, the Netherlands Center of One Health, we are quite proud that one of the members of that committee was a founding member of the NCOH, Marion Koopmans, who can be seen on this slide. So well, we know that the source is in animals, but the question we address here is what is their role in the spread now? And in the in the literature, which obviously you will follow, you can see that well, a couple of studies have been published, or quite a few studies have been published already on the infection of COVID-19 or SARS-CoV-2 in animals. These include natural infections like that have been shown in dogs, cats, but also tigers made the news in the Bronx, but also in Sweden more recently. Lions, mink, ferrets, and the gorillas in California also more recently, like you see in this graph or this picture given, which I need partly to give the credit to Els Broens, one of the next speakers, for coloring in the monkey and the gorilla on this slide of Golzar and Hossein. Also, experimental studies have been conducted, so quite a few species appear to be susceptible to experimental infections, again, including the ferret, cat, the dog, fruit bats that have been tested in Germany, the hamster, obviously used a lot in labs as an experimental model for humans, rabbits, raccoon dogs, mink again, like also from the natural infection, also monkeys, and well, cattle appeared to be susceptible as well to experimental infections. And the pigs I've reported here between brackets, because most of the studies in pigs have not been successful, but one Canadian study was able to infect a few pigs. At least they demonstrated that in EID recently. So that's at least the susceptibility for infection. And then, of course, it's important to know whether these animals are capable of transmitting the infection. And that has so far been demonstrated in the species given here. So ferrets, obviously used as an experimental model a lot. In cats, a couple of studies have been reported or shown that cats can transmit the infection amongst each other. Hamsters, and again, the mink, which were also able to do that in the field. So that is at least showing that some of the animals that we encounter are able of at least having a replication of the virus upon inoculation. But well, what would it mean? SARS-CoV-2 is efficiently transmitted between humans. We see when we follow the daily updates of the Johns Hopkins University, like you see here for yesterday. We've already 112 million people proven infected, as is given in this on this slide. Maybe few of them have been caused by animals. So why bother about the transmission in animal populations if the infection is efficiently transmitted between humans? Well, part of the answer in that is maybe given in the outbreaks that we've studied in minks. You've probably seen in the news that SARS-CoV-2 outbreaks in minks have occurred in several countries. Well, in Europe and in North America. And in the Netherlands and Denmark, these resulted in huge epidemics, including, well, in the Netherlands, some 70 farms, and in Denmark, over 200. The epidemic graphs are shown in this slide, and also the distribution across the country. So these were outbreaks going from farm to farm that were very hard to stop. So these outbreaks indicate in the Netherlands and Denmark that, well, besides humans, kept animal populations can act as a reservoir of the virus. So these animals have clearly shown that in their habitat, in the situation where they are kept, they are able to maintain the infection, have a reproduction ratio bigger than 1. So the infection is not going to go away spontaneously. What is also clearly demonstrated by these two countries is that infection control in a two-host species transmission system, so in this case, which has both humans as a replicating host and mink as a replicating host, it's much more complicated to control than if you are just dealing with a one-host system. So if it's just minks or some other animal species that is infected, and that is, of course, because the infection can go from human to a mink. Then the minks, they stay on the farm, so they do not transmit the infection to another mink or to a mink on another farm, but they may infect a human, as we've demonstrated. And then this human can go to another farm, and in this way, in this two-host system, the infection can be perpetuated and being very difficult to control, much more difficult than in an animal disease where through biosecurity measures you could stop the infection. And what we've seen in the Danish example is that mutations of SARS-CoV-2 in animal species may lead to virus variants of public health risk, which is, for example, the cluster 5 variant in Denmark. And this has led to both countries depopulate all mink farms to stop the outbreak. That was the only solution that in the end could be thought of. So indicating that really is a problem. And then at least in the Netherlands, people didn't think that was that bad for mink farming because they would have stopped anyhow in 2024 because of welfare reasons. But well, suppose you get an infection like that in some other animal species, cats in particular. Most of our disease control systems in animals, they are really tailor-made or made for farmed animals, whether it's avian influenza or foot and mouth disease. But what if the other animal species would be a companion animal like a cat? What would you do then? And that's actually the topic of investigation of this project, which consists of 6 work packages. The first one is on cats and dogs in households and in other populations, and you will hear about them from Herman Egbrink and Els Bruns. The third work package is focused on the pathogenesis and the transmission of SARS-CoV-2 in cats. This work package is led by Professor Wim van der Poel from Wageningen Bioveterinary Research, and we don't have the results yet, but its aim is to not only look at direct transmission between cats, because that has already been studied elsewhere, but it's still important to have the rate of infection from cat to cat, but also to look at the infection that is induced by exposure of cats to a contaminated environment. So if you have an environment that has been contaminated by infected cats and you bring in new cats, what is the probability that they get infected? Just to simulate the indirect contacts that cats may have among each other. The Erasmus Medical Center is also participating in this project and doing the molecular epidemiology, trying to figure out what is the similarity between the viruses that have been found in cats with those that have been found in humans, in order to be able to at least infer on the transmission and maybe also on the direction of the transmission. And then the final part of this project is based on trying to combine it all together, a work package led by Professor Mart de Jong of Wageningen University, where the results of each of these work packages will be combined to kind of estimate the potential role that cats play in the transmission of SARS-CoV-2. It's a bit shown in this picture, where you have various households that may have cats or may not have cats, and you have a virus that may be transmitted within the households from humans to cats, and maybe also from cats to humans, but could of course also by the cats be transmitted from one household to another household. So using the transmission parameters estimated from the work packages and the incidence of infection from the work packages, this is combined to this two-host transmission model of humans and cats, trying to quantify the transmission, the reproduction ratio in humans and cats combined. Results of this project are expected before the end of this year, so I cannot tell about that, but would like to conclude this presentation before I hand over again to Maarten and Els and Herman, to thank the participants of this research, of course our funder, the Ministry of Agriculture, Nature and Food Quality, but also the other NCOH partners involved here, Mart de Jong of Wageningen University, Wim van der Poel from Wageningen Bioveterinary Research, Marion Koopmans from Erasmus University Medical Center, and of course many of their co-workers that I could not give here on this slide. And having said that, I give the word back to the chair.
Thank you, Professor Stegeman. There's no questions in the chat yet, so a reminder to all participants that you can ask your questions in the chat. And I guess because of that, unless Dr. Bruns or Dr. Egbring has a question for you right now already, I would suggest we continue with Dr. Egbring. Let's see, I think we just probably just move on and come back to you later. So our next presentation is by Dr. Herman Egbring. He's a veterinarian and works at the Faculty of Veterinary Medicine, Utrecht University at the Department of Virology as associate professor. His fields of expertise is in molecular virology, diagnostics, vaccine development, and antiviral therapy. Dr. Egbring will tell us more about the detection of SARS-CoV-2 infections in companion animals. Dr. Egbring, the floor is all yours.
Okay, thank you for the introduction. I'm happy that you did not tell my anniversary in the many years that I'm working here. I first try to share my screen now, which should, yeah. I hope you see it.
It's starting up, yeah, there it is.
Yeah, okay,
yeah.
Great. Then I have to, yeah.
Perfect.
Okay, great. So as introduced and also mentioned by Arjan already, a major part of this fighting COVID-19 project is also on studies of SARS-CoV-2 infections in companion animals, and then especially in dogs and cats. And I would like to show you some of the data that we have so far, and I will mainly focus on the development and validations of the tests that we used to screen companion animals. And in the next presentation, Els Bruns, she will show you some preliminary results. So let's first to this study in cats and dogs. I think it's clear and evident that these studies are important. We need to know more about the infection, the disease, and the clinical course. Also from a veterinary perspective, what is the role in cats and dogs? Get more data on the prevalence of infections, as already mentioned by Arjan, in different animal populations. And if we think of cats and dogs, that means populations like catteries, but also in domestic situations, household cats and larger animal populations. Study the possible modes of transmission and also determine the risk of animals becoming a reservoir for SARS-CoV-2, especially also in cats. And this might be important at the time that the number of infections in humans will decline. And of course also determine the strategies to mitigate the risk of infection if needed, if we find a lot of infections. So I decided to just briefly summarize also a few data on experimental infections and natural infections that have been reported already and have been published already to show you what is known about, what we know now about the process of infection in cats especially, and in dogs. This is a study by Gaudreau et al. And in this case, I'm not sure if my pointer also works, I hope so. So in this case, 2 groups of 3 cats were involved. They were infected with a strain of SARS-CoV-2, and 1 day later, or 1 or 2 days later, an extra cat was put into the same cage as to study contact transmission. And subsequently, these cats were necropsied so euthanized and necropsied at day 4, 7, and 21 days post-challenge. And then the tissues, etc., were screened for presence of virus and also for histological lesions. So I will show you just one of the results, which I think is important also for the understanding of the rest of our presentations. And this is the results of the viral RNA load that they found in different samples, in this case in nasal swabs, in oropharyngeal swabs, and in the rectal swabs. And as you can see from these graphs, already 1 day after infection, 1 or 2 days after infection, these samples become positive for virus in the nasal swabs and in oropharyngeal swabs. And then you get a steep decline of the number of virus RNA or virus particles, and virus shedding remains until 7 to 10 days. So then the cats seem to cope with the infection, and that also coincides with the development of very virus-specific antibodies, which is can already be found at 7 days post-challenge. So also the contact cats are infected, so cat-to-cat transmission has been proven also in this experiment. Already also after 1 or 2 days after they come into contact with a positive animal, and also their shedding stops at 7 to 10 days post-infection. Of note is that the shedding in the fecal samples, it stays for a bit longer, or remains a bit longer, takes a bit longer, approximately 14 to 21 days post-infection. In the experimental infections, and that was seen in most of the experimental infections that have been done so far, which are about 3, I think. These cats also don't show any obvious clinical signs, and on abduction, only mild to moderate histological changes can be found within the airways. There's also one study done in dogs, so there are less studies available for experimental infections of dogs. This is a study from Xie et al. He also, or she actually, I don't know, he inoculated also cats, but with more or less similar results as in the previous experiment. But here he also included pigs, chickens, and ducks, which were not infected in this experimental setup. But in dogs which were inoculated, No virus was present in the oropharyngeal swab several days after infection, as you see in this table. Interestingly, some of these dogs became positive in the fecal samples, and 2 of these dogs also seroconverted, which is evidence for the infection, of course, in this experiment. Also, the contact animals, so animals that were brought into contact 1 day or 2 days after the infection of the inoculated animals, also these animals did not become infected. So the conclusion was dogs are less susceptible, and also no dog-dog transmission was observed. So that's just very briefly a few data about the experimental infections. And if we look at the published data so far on natural infections, then you are probably familiar with this first case of SARS-CoV-2 in a dog from Hong Kong. This dog was housed in a facility where dogs that were from owners with COVID positive dogs were kept for longer times. And this dog became was positive for 12 days, and then got negative again. In the same facility, another dog was found after a few weeks to be positive. And then the first cat was detected in Belgium, found to be positive. This cat had some clinical signs, although it was not clear whether this was really due to the infection. And interestingly also, and you're probably familiar with that, is that there were also some infections in big felids, exotic felids, like the lions and the tigers in the Bronx Zoo in New York. And just to summarize some of the other data, so the reports so far are always of sporadic cases of SARS-CoV-2 positive animals, at least virus positive animals, mostly done by PCR. And in most cases, there is a clear connection with COVID-19 positive owners. So the presumed infection is also from their owner. Seroprevalence studies have also been published already. And later on, you will see some preliminary data on prevalence studies done within this project. But human to companion animal transmission, the data on that are quite variable. And there are studies that show no transmission at all. And an example of that is a study that was done in a veterinary student home or veterinary student community, where several of the students were positive for SARS-CoV-2 and several also had clinical signs. And as you can expect, in these houses, there are many cats and dogs living as well in close contact with these students, and there was no transmission at all. And the 15% is from a study from Wuhan, also looking for antibodies in cats taken during the period of the pandemic, height of the pandemic. And 15% of these samples were shown to be seropositive, and some of them with known COVID-19 exposure, at least owners with COVID-19, and some of which it was unknown. Also here in natural infections, often you see no or mild clinical signs, especially in cats. And most of these studies, they study cats. And so far, there is also no report of any transmission event from dogs and cats to humans. Okay, so that was just briefly summary of data from the literature on natural and experimental infections. And now I want to introduce the Fighting COVID-19 project, and especially the studies that we are doing on the prevalence of SARS-CoV-2 in dogs and cats. For this, we use the PCR to detect virus, and we use actually 2 targets, the E gene, which is also used in human diagnostics, and the RDRP gene. And we developed and validated our own SARS-CoV-2 specific ELISAs, which I will show you in a minute. And of note is that in animals, also in cats and dogs, you have several other coronavirus infections, which is of course important if you develop a diagnostic assay, because they need to be specific. In dogs, for instance, we have canine and feline coronaviruses, alpha coronaviruses, which are causing endemic infections in these species. And in dogs, there is also known another beta coronavirus, the respiratory coronavirus, which belongs to the genus beta coronavirus, similar as SARS coronavirus. So what we use for these ELISAs, for these studies, is the S1 protein and the receptor binding domain of SARS-CoV-2, and S1 because it is the part of the S protein, so the outer membrane protein that binds to the receptor, to the H2 receptor, and the S1 part is the least conserved part, and therefore, well, likely a potential target for coronavirus type-specific antibodies. Also, the N protein was included. It's sometimes also used in the ELISAs for humans, for screening antibodies in human sera. And we set up the pseudotype neutralization assay, or neutralization assay with a pseudotype virus, with the vesicular stomatitis virus bearing the SARS-CoV-2 S protein, because we used the neutralization still as a gold standard to confirm positive sera. So what were the results that we obtained? So we used as a source of possible positive sera from the cats from the mink farms, because we expected that many of these cats might be infected and might be seropositive. And we had one serum from a dog from a COVID-19 confirmed household. So what you see here on this slide is to the left is the RDOD values that we obtained with the S1 and with the RBD protein. And indeed, we found 10 out of 44 cats being positive in the ELISA, and also the dog was positive. Also recognizing the N protein, so the nucleocapsid protein, and they were also positive in the neutralization assay. This is even better visualized in this heat map that you see on the right of this slide. In the heat map, in the upper part of the heat map, you see all the positive samples recognizing S1 and RBD both, and also all positive in the virus neutralization assay. And there was a very good correlation between these two proteins in ELISA, and also between the ELISA and the neutralization data. SARS-CoV-2N was positive. Most cats were positive. However, we found two samples that were also negative in all the other assays, but seemed to recognize the N protein of this SARS-CoV-2. So this might be due to cross-reaction, which I will show you later, cross-reaction with other alpha corona or other coronaviruses in cats. So to look for the specificity of the ELISA, and we included sera from the pre-COVID period, 45 cats and 45 dog samples, so sera before the COVID period. And there we saw indeed that what we of course expected, they were negative for S1 and for RBD, but some of these were also positive for the N protein. And we took along the feline coronavirus type 1 S1 protein, which is the alpha coronavirus, just to show that these sera were still reactive. And many of these cats, as you see, have antibodies against feline coronavirus S1. These are the data from the dogs, 45 dogs. Also, one dog was positive for the N, although his OD value was just above the cutoff value. But interestingly, also is this reaction against the bovine coronavirus S1, which we used as a proxy for the respiratory coronavirus, so the beta coronavirus in dogs. And as you can see from these data, none of these samples recognize the S1 and the RBD of SARS, showing that this is quite specific, and that you don't get cross-reaction if dogs are also infected with the other beta coronavirus, the endemic beta coronavirus in dogs. So we concluded that this ELISA was quite specific. We still had to solve that reaction with the N protein, whether this might be due to the cross-reaction with, for instance, feline coronavirus, the alpha coronavirus. And for that, we screened SPF cat sera that were available from previous vaccination studies in our lab. And as you see here, the sera of the infection, and they do not contain, of course, as expected, antibodies. Post-infection, several weeks post-infection, these cats do develop antibodies against the S1 of the feline coronavirus, and also several of these cats are positive for the N protein. So this clearly shows that this reaction is indeed due to the cross-reacting antibodies in these cats, and that the N protein cannot be used, at least for the screening of cat samples and dog samples. And you can also already depict that from looking at the amino acid identity of these proteins, where you see that the nucleocapsid protein has a higher percentage of amino acid identity as compared to the S1 and the RBD. So to conclude and to summarize, for this part, we have developed this S1 and RBD ELISA and shown to be quite specific, and that it can be used for screening of large numbers of samples from cats and dogs in the study that will next be presented by Els Bruns. And as a criterion for a positive sample, we use the ELISA, but still we do virus neutralization to confirm the positive samples. And it was clear to us that the N protein ELISA could not be used due to cross-reacting antibodies. And with that, I would like to conclude, but not before I have shown you these acknowledgements, and especially acknowledge Sansao and Nancy Schuurman, who did all the serological work that I presented today. Thank you.
Thank you, Dr. Eckering. Well, there's a bit of an echo. Apologies for that. There's so maybe if Dr. Eckering, we can switch mute and unmute, otherwise it's hard to follow people.
Should I stop sharing?
No, this is mute while I talk, and then I mute when you answer the question. Does this work? This works better. Excellent, because the echo is quite annoying. Thank you for your presentation. I recognize the cats. The picture of the dog I didn't recognize as a dog. You sure there was a dog? Okay. There's also a question we got from Barry, saying that assuming the contact with lions, tigers, etc. is quite limited, and hopefully at a safe distance, is anything known how these infections have occurred? Did animal caretakers, were they found positive, or any indication of infection from contaminated foods, or how do you suppose this infection came to be?
Yeah, there have been, of course, several suggestions and hypotheses about the how these animals became infected, and the most obvious one is, of course, that some of the zoo caretakers were animal caretakers, that they were have transmitted the virus to these animals, and that was certainly the first theory. But as far as I know, it was actually never proven that they had been positive. And then, of course, the next theory was that it might have been transmitted via food. That might also be the reason, because it's obvious that it's not a direct contact with people. It should also always be indirect. And the zoo was closed at the time, so it could also not be visitors that had transmitted the virus.
Thank you. I think for now, these are the questions from the chat function. There's a few others for general, which we'll repeat later. A reminder to all participants to ask your questions through the chat. Apologies. And then Dr. Eckering, we now move on to Dr. Els Bruns. Dr. Els Bruns is head of the Veterinary Microbiology Diagnostic Centrum, or VMDC, of the veterinary faculty. The VMDC is part of the Department of Biomolecular Health Sciences. Dr. Bruns is a specialist of veterinary microbiology with a PhD on MRSA in pig farming. Her key interests are in clinical microbiology, antibiotic resistance, zoonoses, and One Health. Dr. Bruns will tell us more about the preliminary results of the study fighting COVID-19 in companion animals. Dr. Bruns, the floor is all yours.
Well, thank you, Maarten, for introducing me, and thank you, Arjan and Herman, for, well, telling all these things that I can go on with, and, well, for me, the honor to present the preliminary results of whatever it's share. So yep. I'm sharing now, yeah. So the honor to me to present the preliminary results of this project we've been doing, and yeah, Arjan already told all about the whole project. I will focus on work package 1 and 2, and Herman told you all about the tests we use for this project. So work package 1 is on SARS-CoV-2 in cats and dogs in Dutch households, and it exists of 2 tasks. One is what we call the household study, and the other one is the vet study. Well, so task 1 is the prevalence in cats and dogs in positive households, and task 2 is the prevalence in cats and dogs visiting the veterinarian. Well, let me see. The first one, the household study, well, we did that first. We aimed to do that in collaboration with all kind of projects in April and May last year, which were on the human side. But when we started off, well, the first wave was going down, so there were not a lot of people and participants in these projects. So then when the second wave came, we asked the municipal health services to ask people who were tested positive to inform them about our project, and then these people could contact us, and then we will make an appointment for a house visit. But as these people were COVID positive, of course we cannot just go and visit them, sit on the bench and cuddle with their animals. So we had to think of how to do this. And then we came at the Stray Cat Foundation in the Netherlands. They have this nice van that you see, Catopia. It's named Catopia, and it's well, internally it's totally like a veterinary clinic. So we could take the animals out of the house, go into the van and sample them. Well, our target was to get 150 cats and 150 dogs, and we took oropharyngeal and rectal swabs for PCR. And just, well, Herman didn't talk about the PCR, but we just used the same PCR as we do on the human side in the Netherlands. And of course, we validated that there was no cross-reactivity with the feline and canine coronaviruses. So, and we took also a serum sample for ELISA and VNT, what Herman talked about, and we took a short survey, and if there were PCR positive animals, we did a follow-up. So here are some preliminary results already there. So in this table, you see first the animal species. This is the first house visit. You see the animal species, cats and dogs. So we included 155 cats and 156 dogs. And you see that we got 6 cats and 7 dogs PCR positive. And if you look at serum positive, so antibodies, you see that this was 29 cats and 18 dogs. And well, if you count it up, well, it's not that 6 plus 29 because some of the PCR positives were seropositive as well. But so we end up with 31 SARS-CoV-2 positive cats and 24 SARS-CoV-2 positive dogs. And so you see that 1 out of 5 of the cats are infected when their owner is SARS-CoV positive, which is quite a high percentage, I guess. And then if you look at, we had 196 households. So these cats and dogs were from 196 households, and 20% of the households we found positive pets. So then we did follow-up visits, as I said, from the PCR positives. Well, these were 6 cats and 7 dogs, but we included for the follow-up only 6 dogs and 5 cats, as the owners didn't want to, well, they didn't want us to visit again. So and so we did this 11 animals, and also the animals who were in the same household. So sometimes they had 2 or 3 cats. So we sampled these again, because maybe there was some transmission within the household between the cat and the dog, or between cats. But we didn't find any new PCR positive animals after the first house visit. So it appeared that there was no transmission between the pets in the household. after our first visit. And what we saw is that all PCR positive animals were or became seropositive, because of course PCR positive can mean that they only are contaminated because they have some, maybe have cuddled with their owner and have some of the virus, but they became seropositive. So that's where we concluded there was an active infection. And apparently we found 3 cats from 3 different households which were still PCR positive at the follow-up visit, so after 1 or 2 to 4 weeks. And 2 out of these 3 cats we sampled again, so for the 3rd time, and they became negative. And the other, the owner said it is enough, the cat doesn't like all this sampling. So, well, I assume the cat turned negative again, but you can see that there is maybe a little bit longer time of PCR positivity in these cats. As Herman already mentioned, that in the literature also there's no or mild clinical signs reported, and we asked all the owners of the positive tested pets, and they didn't report any alarming signs. So maybe some, if you ask them, said, oh, maybe I saw that he sneezed a little bit, but well, it was more seeking for signs than that there were really signs. What we also saw that oropharyngeal swabs were more often positive than rectal swabs. And the PCR positive animals in the household were households with very recent COVID-19 infections. Because of our logistics, so we had this van and we had to do route tours through the country. So we had like a tour in Friesland and then a tour in the western country, part of the country. So sometimes we were there very recent, or sometimes just like 1 or 2 weeks after these people were out of quarantine. So what we see is that these PCR positive animals are only then when there's a recent infection. And what we also saw is that in households with more than 1 pet, that often only a proportion of the pets was positive. I know that's in humans the same. Sometimes all people get it in a household, but sometimes it's only 1 or 2 people. So that's what about the household study. Then we have the vet study, where we have 30 to 40 veterinary clinics working with us. And we asked them to send in 20 samples, 10 from cats and 10 from dogs, and then 10 from animals with signs, like clinical signs like respiratory or maybe diarrhea, vomiting, which mostly is seen with coronaviruses, with the normal coronaviruses, and 10 animals without signs. Again, the swabs for PCR and serum for ELISA and a short survey. Well, this study is still running, but so far we have 109 cats and 152 dogs. We only have 2 PCR positives, 1 cat and 1 dog. And when we phoned the veterinarian, they appear to be both living in COVID-19 positive households. We had a little bit more seropositives, 4 and 2, but the percentages are much lower than in the COVID positive households. Then there's work package 2. Well, actually, Herman is the project leader, but the honor is on me to present the results. I will present from 2 studies, the serum bank study done by Xian Zhao and the shelter cat study coordinated by Rut van der Leij. There we did a seroprevalence in serum bank samples. That's what how we call it. Maybe you can a little bit compare it with what the blood bank did in the first wave. What we used is 1,000 sera submitted for routine diagnostics to labs from the Faculty of Veterinary Medicine for other kind of diagnostics. So we don't know anything about the COVID-19 exposure, and we used the ELISA Herman already told about. So, and then you can see here in the table that we had 500 cat samples and 500 dog samples. And again, the S1 ELISA, the RBD ELISA, and the virus neutralization test, which was done on the positive samples and on the selection of the negative samples. And in total, we had our definition of seropositive was when all 3 tests, so S1, RBD, and VNT was positive. And then you see that in cats, there are only 2 out of 500, so 0.4% seropositive. And in dogs, it was just one dog, seropositive, so 0.2%. In cats, we saw something else where we had S1 and RBD positive cats, but the virus neutralization test was negative. So 0.8 were inconclusive, and we are now looking into these samples to see if we can clarify what is happening here. So, but here the prevalences are very low. These are samples from April and May 2020. So it was during the first wave, but probably if we will repeat it now, maybe we get higher. So we are planning to repeat this study again. Well, maybe on the right side, you see the map of the Netherlands, and you see the geographic distribution of the cat samples and the dog samples. So it's pretty much over the whole country, but a little bit centralized by Utrecht. That's because of most of the veterinarians from other parts of far will maybe use other labs than the Utrecht labs. And then I said there's the shelter cat study where we wanted to look in the shelters because this is a total different population of animals who live in households. In shelters, these are stray cats maybe and other kind of cats, and they're mingling in groups sometimes. So that's why we wanted to look at the shelter cats. And well, even we wanted to see, and well, maybe we still hope for the opportunity to have an outbreak in a shelter to follow up that outbreak to, we can do that experimentally, but we would like to see it in nature as well. But so far we don't have an outbreak in the shelters. As I said, Rut van der Leij is coordinating this task, and she contacted 30 shelters. We want to have 10 cats per shelter. Again, we did only serum, and then if there were seropositives, we did a PCR from seropositive cats. So far, we have 251 sera from 28 shelters, 2 cats seropositive, and both cats turned out PCR negative. Sorry, I get some text messages that the internet is, but I think it's not my internet, but I hope it's still working. So preliminary conclusions of these 2 work packages is that pets are only PCR positive after exposure with COVID-19 positive humans. There's a very low seroprevalence in pets with unknown COVID exposure, so less than 3%. A higher seroprevalence in pets with COVID exposure, 15 to 20%. We see no or mild clinical signs in these positive pets. And so the most likely transmission route seems to be from human to pet. Pet to pet transmission is less likely, might be within a household, but between households, especially between cats are normally, do social distancing by nature. So less likely, and pet to human transmission not likely is what we think. So then based on these preliminary results, there were some recommendations and precautions changed. Of course, this was done by the National Institute for Public Health together with us. And so for COVID-19 positive persons, now the advice is to avoid contact with your pet, do not cuddle them, and do not let them lick you. The latter one I would advise for always. So, keep dogs and cats indoors as much as possible during isolation or quarantine period and or that of your household members. And only walk dogs for a short time and on a leash, and arrange for someone in the household who is not ill to take care of the pet if possible. And for veterinarians, which was circulated by what we call a vet infect, which is like a message for all professionals in the veterinary profession, is that you have to use appropriate protective equipment when pets come in from COVID-19 positive households, because so far we haven't seen transmission from pets to humans, but we really want to avoid that for happening. So that's why we have these recommendations and precautions. So that's, again, maybe summarizing the partners and the funder of our project. Thanks for that. And that's my last. So if there are any questions, maybe you have done it in the chat. I haven't seen it during my presentation, but I'm sure Maarten will have looked at the chat. And I want to thank all these people, and probably I forgot some, but there are a lot of people involved in these projects.
Thank you, Dr. Broens. Very interesting, and I'm glad to hear that the risk seems to be limited to us, and certainly not an animal reservoir of cats and dogs to the COVID-19. Is that correct?
Well, based on these preliminary results, I think it's limited. Yes, and so I think that the danger or the risk is limited as well. But maybe Arjan, who knows a lot about the epidemiology and the reproduction ratio, might disagree or might have some more well, we can't conclude that yet, but that will be done in work package 3 and 5 in the project. So, but maybe Arjan wants to add something to that.
Yeah, the point is that I think it's too early to conclude, Martin, and the reason is that, well, it's the combined transmission system that might that is of relevance here. So yeah. we see the estimates of the reproduction ratios among humans presented each week here in the Netherlands, and then, well, they are something like 0.9 or at least around 1. And maybe for cats, also, if it would be quite modest, maybe 0.2 or 0.3, then the combined reproduction ratio of the system could just tip it over 1 or below. So in that respect, it might be relevant. An interesting veterinary example of such a two-host system is, for example, bovine tuberculosis in Great Britain, where you have two populations that are affected by bovine TB, the cattle population, which is classically infected with bovine TB, and the badger population. And from estimation, it follows that in both these subpopulations, the reproduction ratio is below 1, so the infection should fade out. But the combination of the two actually makes a combined system where it's able to perpetuate and to continue itself. So in that sense, also the combination of the two in the modeling work package 5 will actually reveal whether we could support Els in her conclusion or not.
Yeah. Dr. Ekbrink, you wanted to add something to this?
Yeah, maybe it's also good to mention that when evaluating these results, and we had these discussions before, you also have, of course, to take into account the behavior of cats and how we keep our cats. So normally in a domestic setting, there are only maybe 1, 2, or 3 cats in a household, and knowing that the shedding of virus is not long lasting, I would expect that in these situations the virus will die out, and then of course it's different from a population that has continuous contact with each other. And cats don't have that, but it will be interesting to see how the model works out with input of all these data that we will generate.
Yeah. And is it possible for a dog to get infected from eating cat's excretum? Because dogs love eating cat excreta. I mean,
I think you cannot exclude that. What we find in feces is viral RNA. What it is infectious virus, we have not looked at. It might not be infectious, so in that case, it of course will not be infectious for the dogs as well. But the risk, I think, is not that high. In a household, that might be one of the sources of transmission, not only eating but also licking. Outside, cats are normally burying their feces. So then also the risk, I think that also makes the risk of transmission less likely.
Yeah. That relates to a question that was posed by one of the viewers, is why did you actually decide to take rectal samples? Do humans' rectal swabs also become positive if they have COVID-19?
Well, the last question about human samples, I cannot answer. Probably yes, but I think we chose that because of the experimental results first, because we had first the results of the experiments in Wuhan, that they had rectal swabs quite positive with quite high or low CT, so high viral RNA in rectal swabs and in feces. So and we thought that if feces is a good proxy for being infective, yes or no, then it's much easier than to take oropharyngeal swabs. But well, it turned out to be in this study, which had only, that I said, 13 PCR positive samples, that the oropharyngeal swabs were much more positive than the rectal swabs.
Yeah. I can add to that there's a lot of sewage studying going on for populations, city populations, which pretty well predict the epidemiology of COVID-19 within the human population. So the answer is yes, humans also excrete COVID-19.
Maybe we can change this test strategy in the test streets, that you don't have to go in your throat, but another swab taken, maybe.
Yeah, but perhaps it's not that good, because I can imagine, like Martin is saying, that on the population level you are able to follow the infection. But I think in most species, also, for example, if you look in mink, then the proportion positives in throat samples is much higher than in rectal samples, like in cats. And if you look at CT values, then it's 10 cycles difference, at least in the mink example. So apparently throat testing is still much more sensitive. So we might let a lot of people on the streets that are infected else, if we have them present their stools.
Yes. And it will be hard to place people into quarantine individually based on joint sewage samples. Back to cats and dogs. There's a few more questions that are partially answered already in the chat, but I'll repeat them a little bit. A question by Rolf Nijssen. Do you have any data on ferrets that are kept in households? Were they positively tested?
No, we have like one clinic in the Netherlands which is specialized for ferrets. There will be more, but this is called the ferret clinic. So this veterinarian was very willing to cooperate, but then she became ill herself. So we didn't get any samples so far. But we will try to, if she's better, then we will try to incorporate her. I know, but Herman probably can, or Arjan can add that. I know that there are in another country, there are some pet ferrets turned out positive.
Yeah, there are not many data on infections, natural infections in ferrets, but there was one now from Spain. And actually, I think that was the only one so far.
I think it's one additional, Herman. So one from Spain, which I think was only serology, and then also one, I think, from a Balkan country, which also was PCR positive.
Yeah, might be. Yeah. But not many natural infections, although ferrets are quite susceptible, and at least under experimental conditions, then you can easily infect them. Yeah.
But under experimental conditions, cats are also quite easy to infect, right?
Yeah.
So do you think there's discrepancy between how easy it is to infect a cat in a experimental setup and how the level of seroprevalence you see in domestic cats?
Well, it might be due to the of course the strain of the virus that is used for the experimental infections, and also of course also the affecting dose that you use for the experimental infections, which is considered quite high. And maybe also the age of the animals. That in one of the experiments, actually the first one that was reported, that was published, that was the experiment done by sheep. What they saw in their experiments is that especially juvenile cats were also more susceptible, and these cats did also show some clinical signs than the older cats. So these might all be factors that can explain the differences that you find between the experimental infections and natural infections.
Yeah. Back to the cat shelters, because that is really interesting indeed. Maybe you will see an outbreak. But the question from Annemiek is, how were the shelter cat samples selected? Were they selected from the serum bank, or how did that work?
You want to answer that, Herman, or shall I do that?
Whatever.
Okay, I can answer that. No, it were so the shelters were contacted to sample for cats who are in the shelter at the moment. So these were not serum bank samples. These were, so we have some data on the background of these cats as well, but we didn't analyze these yet. So I don't know, because I saw some other question of if we didn't know where the serology positive cats came from, whether these were stray cats, yes or no, but that's what we don't know yet, or we didn't analyze it so far.
Well, so that I just asked that, to be honest, yesterday to Rut, and so you couldn't know that. But these two seropositive cats from the shelters were indeed abandoned cats, so they were not stray cats. But whether the owner had COVID or not, that's unknown. Yeah, so it's maybe also good to add, if you for the selection of these cats in shelters. One of the objectives was also to only get samples from cats that were housed already in shelter in a group housing, so that they did not enter, so not after the first day that they enter, so they had to be in shelter for a bit longer time. Also, whether also in a situation where we would find a positive cat, to see whether there was spread within that group. So that was also the reason why we specifically selected for these cats.
Interesting. Well, it will be interesting to see the results in the other work packages. Unfortunately, the time is already finished. It's 6 o'clock. So Professor Steegman, Dr. Egberink, and Dr. Bruns, thank you very much for your time and your interesting presentations. Our next webinar will be on the 18th of March. Professor Marion Koopmans and Dr. Maria van Kerkhoven will give a talk on COVID-19, the role of WHO, and the recent WHO mission of Marion Koopmans to
Maarten, je moet jezelf even unmuten.
Sorry, I don't know what happened. Apparently I got muted. Where did I get muted?
Just go on and conclude.
And conclude. Yeah, I don't know. Did I talk already about our next webinar?
A little bit, but please share again.
Okay, I really apologize. Don't know what happened. The next webinar will be on the 18th of March with Professor Marion Koopmans and Dr. Maria van Kerkhoven on COVID-19 and the role of WHO and the recent WHO mission to China. Please subscribe to our newsletter. You can contact us on info@ncoh.nl. You can also send suggestions on webinars and topics you find interesting. So since I got muted and unmuted, I don't know if I thank the speakers sufficiently. So again, thank you, Professor Steegman, Dr. Egberink, and Dr. Bruns. I hope you enjoy your dinner, and we're looking forward to seeing more results of this interesting study, perhaps in another webinar in the near future. And with that, we conclude this webinar. Thank you. Bye-bye.